Hey everyone, it's Dr. Marcon, and this is Chapter 12 on the fundamentals of the nervous system and nervous tissue. This is one of our first lecture chapters for Unit 5 going into the nervous system. So just some characteristics about the nervous system. The nervous system is the master control and communication system. There are three overlapping functions of the nervous system. So first, sensory receptors will monitor changes inside and outside of the body. It detects stimuli, or a stimulus are changes that are detected inside or outside the body. Also, we have sensory input. This is information gathered by receptors. So we have processes. The nervous system also processes and interprets sensory input. This is called integration. So integration is a term for basically interpreting and processing input coming from the outside. The nervous system also dictates a response by activating effector organs, and usually motor output is the response. So here we see the basic functions of the nervous system. So first we can, the eyes see a glass of water. Yes, water. The eyes will see a glass of water, and this information coming from the eyes are then brought to the nervous system via sensory afferent fibers. So sensory input coming from the eye is then brought to a part of the brain where integration or processing occurs. So this information is processed, and then the brain will basically tell the body what to do. So it will send a motor signal. So the motor output will then go to the effector, which is in this case the skeletal muscle within the arm. So the arm will grab the glass of water and give the person a drink. So we have basic divisions of the nervous system. The first division is the central nervous system. Now there are only two main components of the central nervous system. The central nervous system is composed of the brain and the spinal cord. So the CNS is the integrating and command center. The next division of the nervous system is the peripheral nervous system, also known as the PNS. The peripheral nervous system consists of nerves extending from the brain and the spinal cord, and these are the cranial nerves, so we have 12 cranial nerves, and the spinal nerves. The peripheral nerves link all regions of the body to the CNS. And then we have special structures called ganglia. Ganglia are clusters of neuronal cell bodies, and these are found in the peripheral nervous system. So here we see the basic divisions of the nervous system. We have the central nervous system, or the CNS, made up of the brain and the spinal cord, and then we have the peripheral nervous system made up of the nerves and the ganglia. So we have sensory input and motor output. So sensory or afferent signals are picked up by sensor receptors, and these are carried by nerve fibers of the peripheral nervous system to the CNS. Our motor or efferent signals are carried away from the CNS, and our motor efferent signals innervate muscles and glands. So muscles and glands are basically the effectors, or what are going to produce the signal coming from the CNS. So sensory input and motor outputs are divided according to the region which they serve. So we have the somatic body region and the visceral body region. So the somatic body region consists of structures that are external to the ventral body. In other words, structures of the outer tube, such as the skin, skeletal musculature, and bones. The visceral body region mostly consists of the viscera, and we know that viscera means organs. So viscera within the ventral body, which means structures of the body's inner tubes, such as the digestive tube, the lungs, the heart and bladder, and other organs. So this scheme will then result in 4 main subdivisions of the peripheral nervous system. So we have the somatic sensory division or subdivision. This is the sensory innervation of the outer tubes, such as the skin, the body wall, and the limbs. We then have a visceral sensory subdivision. This is the sensory innervation of the viscera. We have a somatic motor subdivision or voluntary motor. This is the motor innervation specifically of the skeletal muscles. And then we have a visceral motor subdivision, also known as the autonomic nervous system. So this is our involuntary motor innervation, specifically of smooth muscles, cardiac muscle, and glands, as well as other structures like the erector pili muscles in our skin, smooth muscle in the vessels, and our sweat glands. So we'll get into detail in these different subdivisions later on. So just generally speaking, when you see a subdivision that's sensory, basically these are our inputs. We're getting information from the outside environment or internal environment. Somatic sensory, these are, our inputs that we're getting with regards to touch, pain, pressure, vibration, temperature, and proprioception, basically things around us. This is coming from our skin, our body wall, and our limbs. Also, our sense of hearing, equilibrium, and vision. So these are inputs that are going towards the brain. Visceral sensory, these are inputs coming from our viscera, from our organs. So we have stretch, pain, temperature, chemical changes, and irritation in the organs, as well as when we have nausea or hunger. Other special visceral sensory information we get are, from our taste buds as well as smell. So sensory, these are our inputs, okay? So this is information we're getting towards our brain. Motor, these are our outputs. This is our brain telling the rest of our body either, what to do, or either voluntarily or involuntarily, okay? So sensory, these are our inputs. Motor, these are the outputs coming from the central nervous system. So here is just a schematic of the levels of organization in the nervous system. Again, we just have the 2 main components of the central nervous system, the brain and spinal cord, and these are our integrative and control centers. We then have the peripheral nervous system, or the PNS, made up of our 12 cranial nerves as well as our spinal nerves. This provides communication lines between the CNS and the rest of the body. So we have our sensory or afferent division. Again, we have somatic and visceral sensory. So this is stimuli coming from our body, either through our skin or through our organs. And our sensory afferent division will conduct impulses from the receptors throughout our body to the central nervous system. So, it goes up the sensory afferent nerve to the integration center within the central nervous system. Also part of the peripheral nervous system is our motor or efferent division. These are signals that are going away from the central nervous system. These are signals that are made by the integrating centers in the brain, so that it can have an effect on the rest of our body. So we have our motor or efferent division. These are the motor nerve fibers, conducts impulses from the CNS to the effectors. So the effectors can either be muscles or glands. And we have 2 different divisions of the motor efferent division. So we have the somatic nervous system. So somatic, this is our motor or voluntary motor division. So we have impulses being conducted from the brain, from the CNS to skeletal muscles. So basically, if I want to use my finger to click on my mouse like that, which I'm going to not do again, I'm using my somatic motor fibers that are coming from my brain to do that. And then we have the autonomic nervous system, or the visceral motor subdivision. This is, again, our involuntary motor division. So this will conduct impulses from the CNS to organs such as the cardiac muscles, smooth muscles, and glands. And this is on an involuntary basis. We're not sitting here telling our hearts consciously to beat. No, this is done through involuntary means, through our autonomic nervous system. And we have various ways of regulating heartbeat, as well as movement of the muscles within our organs. And within the autonomic nervous system, you can then further divide it into the sympathetic division and the parasympathetic division. So sympathetic division being that fight or flight response, which will mobilize body systems during activity, either fighting off the dinosaur or running away. Whereas our parasympathetic division, this is our rest and digest division, helps to conserve energy, also helps promote housekeeping functions during rest. Okay. So the first subdivision that we're going to talk about are the somatic sensory, or our general somatic senses. We have receptors spread throughout our body, throughout our outer tube of the body, that pick up stimuli for touch, pain, vibration, pressure, and temperature. General just indicates that it is widespread or throughout. Okay. We have proprioceptive senses. These detect stretch in tendons and muscles. Basically gives us our body sense, our position and movement of body in space. Like just sitting here, I know that the window is maybe like 2 feet away from me. This is my proprioceptive senses. There's also other special somatic senses, which unfortunately we don't have time to go through, but the special somatic senses include hearing, balance, vision, and smell. Then we have our visceral sensory subdivisions. So general visceral senses detect stretch, pain, temperature, nausea, and hunger. This is widely felt in the digestive and urinary tracts, as well as the reproductive organs. And special visceral senses include taste and smell. So from our inputs, we then go to our outputs, our motor subdivisions. So we have two. We have first our somatic motor subdivision. So we have our general somatic motor subdivision. These are these signal contraction of skeletal muscles, and these are under our voluntary control and is often called the voluntary nervous system. So we are consciously, moving our muscles or telling our muscles what to do. Now the bronchial motor, this is typical skeletal muscle derived from somatomeres. Basically what this means is the bronchial motor, which are a type of special visceral efferent, they innervate skeletal muscle derived from the bronchial arches. This includes muscles of mastication, so the muscles that help us chew our food, muscles of facial expression, as well as the palatal, pharyngeal, laryngeal, trapezius, and sternocleidomastoid muscles. Again, all these muscles are under our voluntary control, so we have voluntary control of them. Then we go to the visceral motor. Visceral motor regulates the contraction of smooth and cardiac muscle. This is what makes up the autonomic nervous system and helps control the function of visceral organs. This is also often called the involuntary nervous system, or what we call the autonomic nervous system. And again, our autonomic nervous system is further divided into the sympathetic nervous system and the parasympathetic nervous system, with our sympathetic nervous system controlling our fight or flight responses, and then parasympathetic nervous system in charge of our rest and digest activities. So now that we've gone through the basic divisions of the nervous system, we'll then get into nervous tissue. So we have cells within the nervous system that are densely packed and intertwined, and we have two main cell types. The first main cell type is the neuron. The neuron is what is responsible for transmitting electrical signals. And then we have our support cells within the central nervous system and the peripheral nervous system. So support cells, these are called neuroglial cells in the central nervous system. Support cells are non-excitable and surround and wrap neurons. So the neuron is the basic structural unit of the nervous system. These are specialized cells that conduct electrical impulses along the plasma membrane. So they basically generate a nerve impulse, and we know this to be an action potential. And you'll learn about action potentials when you get to physiology. Believe me, you will. We have different or special characteristics of neurons. One characteristic is longevity. A neuron can live and function for a lifetime. However, they do not divide. Fetal neurons will lose their ability to undergo mitosis. The only exception are neural stem cells. They also have a high metabolic rate and require abundant oxygen and glucose. Neurons will die after 5 minutes without oxygen. So if you're studying for a really long time, make sure you're getting some exercise, get some oxygen going to your brain, get some glucose, have a snack. Also, if you're performing CPR and a patient has stopped breathing, you need to perform CPR as soon as possible because, again, neurons will die after 5 minutes without oxygen. The first part of the neuron we're going to talk about is the cell body. It's also called the soma. Now, the size of the cell body will vary anywhere from 5 to 140 microns. It contains the usual organelles plus other structures. One structure that we'll see in neurons are what's called chromatophilic bodies or Nissl bodies. These are called Nissl bodies because they have a specific stain called a Nissl stain. They stain blue. It's really pretty. I've actually performed a Nissl stain on brain sections before. Chromatophilic bodies are clusters of rough ER, or rough endoplasmic reticulum, and free ribosomes. They help to function to renew membranes of the neuron. So we know that the rough ER, or endoplasmic reticulum, is responsible for making proteins. And proteins are important to help renew membranes of the neuron. Another structure are the neurofibrils. So neurofibrils are bundles of intermediate filaments. They form a network between the chromatophilic bodies. Now, most neuronal cell bodies are located within the central nervous system. If there are neuronal cell bodies in the peripheral nervous system, these are called ganglia. Okay, so the neuronal cell bodies outside of the central nervous system, again, they're called ganglia. Here we see the structure of a typical neuron, specifically a motor neuron, and we've seen pictures of neurons before in our histo slides. So we have the soma, or cell body, that contains the nucleus. Most neurons have one long axon, and then many dendrites coming off of the cell body that basically receive information from other neurons. We can see in our histo slide the purplish structure, that's the soma or cell body, with a pale colored nucleus with nucleolus in the center. We can see some strands of fibers, which are the neurofibrils, and then the shorter branches being the dendrites. And sometimes, if we're lucky, we can see one really long axon coming off of the cell body. We might be able to see some chromatophilic substances, some Nissl bodies within the cell if it's stained correctly. So we know that the axon actually has these cells surrounding it that help with conduction of impulses, and we'll get into those different types of cells in just a bit. So dendrites are extensively branching from the cell body. They help transmit electrical signals towards the cell body coming from other neurons. Chromatophilic bodies extend only into the basal part of the dendrites and to the base of the axon hillock. These dendrites function as receptive sites for receiving signals, again, from other neurons. A cell body will have many dendrites, however, it will only have one axon most of the time. The dendrites will receive signals from other axons, and usually the axons will attach to the dendrites, allowing for the continuation of the signal from cell to cell or from neuron to neuron. So neurons only have one axon. The axons are what generate and conduct the impulses from the neurons. So we have action potentials that just go along the axons and travel to other cells. Axons will transmit impulses away from the cell body. We have no chromatophilic bodies in the axon, and there's no protein synthesis in the axon. So we have structures such as neurofilaments, actin microfilaments, and microtubules that provide strength along the length of the axon. They also aid in transport of substances to and from the cell body, such as hormones. This process is axonal transport. Branches along the length of the axon are infrequent. There are some axon collaterals, but not a lot of the time. There are, however, multiple branches at the end of the axon. This is known as the terminal arboration or the telodendria, and they end in knobs called terminal boutons or axon terminals. So think of a long axon and then finger-like projections, sort of like a hand, finger-like projections coming off of the axon. These are the terminal arborations, and these terminal arborations will end in button-like structures or knob-like structures called terminal boutons, also known as axon terminals. So nerve impulses are generated at the initial segment of the axon, and they are conducted along the axon to the terminal boutons. Within the terminal boutons, you have chemicals known as neurotransmitters. Neurotransmitters are contained within vesicles, and they are released from these vesicles at the terminal boutons once it gets the signal to release the neurotransmitters when the signal goes along or down the length of the axon. Synapses are the site at which the neurons communicate. Most information is passed through chemical messengers, the neurotransmitters. Some information is transmitted electrically through gap junctions. So you have different parts of the neuron at the synapses. So first you'll have actually two neurons. You'll have a presynaptic neuron, so a neuron before the synapse. This is the neuron that conducts the signal towards the synapse. And then you'll have the postsynaptic neuron. This is the second neuron that will receive the signal. So the postsynaptic neuron will transmit the electrical activity away from a synapse. Okay, so you need two to tango, two neurons to communicate. You'll have a presynaptic neuron and a postsynaptic neuron. So presynaptic before the synapse is what originates or conducts the signal towards the synapse, and then you'll have the second neuron, which is the postsynaptic neuron, which will receive and then transmit the electrical activity away from the synapse. So here you see two neurons connected at synapses. So you'll have one neuron here, and you can see that you have an axon coming from a presynaptic neuron. So here is that axon, and then you'll have those terminal branches with the terminal boutons that will send their signal across, and it will send the signal either to the dendrites or the cell body itself. Here we can see the terminal boutons ending on the dendrite of this one axon. So the presynaptic neuron is the neuron that is transmitting the signal towards the synapse, and then you have the postsynaptic neuron that will be receiving the signal and then transmitting the signal away from the synapse so that it can synapse with other neurons. Okay. And then we have different types of synapses. So we have an axodendritic synapse. The axodendritic synapse is between the axon terminals of one neuron and the dendrites of another. So the presynaptic neuron will send its signal along the axon, and then the terminal bouton will end on a dendrite of the postsynaptic neuron. And this is the most common type of synapse. And then you have axosomatic synapses. This is when you have the connection between two neurons. So the synapse is on the actual cell body. Okay, so the presynaptic neuron will send its signal along the axon, and the terminal bouton will end on the neuronal cell body of the postsynaptic neuron. Okay, so two types of synapses, basically one either from axon to dendrite, the second one from axon to soma or cell body. And here is a picture of a typical synapse. This specifically is an axodendritic synapse, where you have the terminal bouton of the presynaptic neuron connecting with the dendrite of a postsynaptic neuron. So the nerve impulse or action potential will travel down the axon, transmitting that impulse, that action potential, and it will signal the synaptic vesicles to release the neurotransmitter. So here you have a vesicle going to the synaptic membrane of the terminal bouton and releasing the neurotransmitter into the synaptic cleft. Okay, and then receptors on the postsynaptic dendrite will pick up the neurotransmitters and allow for continuation of the signal. And the postsynaptic dendrite is what will carry the message away from this actual synapse. So synapses are elaborate cell junctions. We talked about those synaptic vesicles that have the neurotransmitters. These are on the presynaptic side. In the terminal bouton, synaptic vesicles are membrane-bound sacs that contain neurotransmitters. And we also have mitochondria that are abundant in the axon terminals. The synaptic cleft is basically just the space, and it separates the plasma membrane of the two neurons. So that cleft is that space between the presynaptic and postsynaptic neuron. And then we have different classifications of neurons. We have a structural classification basically based on structure. So you have a multipolar neuron. A multipolar neuron are they possess more than two processes. There's numerous dendrites coming off of the cell body or soma and only one axon. Bipolar, as its name implies, has two. So bipolar possesses two processes. These are very rare neurons, and they are found in some special sensory organs. And then we have a unipolar, also called pseudo-unipolar. So pseudo we know means fake. A unipolar or pseudo-unipolar neuron possesses one short single process. It does start as a bipolar neuron during development, and then at some point becomes a unipolar or pseudo-unipolar. So this next slide actually allows you to see in a table a comparison of the structural classes of neurons. So you have multipolar again, many processes, so many dendrites and one long axon. And then you have the bipolar neuron that has two processes extending from the cell. One is a fused dendrite and one is the axon. And then this unipolar or pseudo-unipolar, typically it looks like there's one process that it will extend from the cell body, but you can see that it kind of branches off. So one end will be a peripheral process and the other will be a central process, and apparently this is one long axon. And you can see the relationship of the anatomy to the three functional regions. Okay, so there are different regions of the axon. You have a receptive region, a conducting region, and then a secretory region where neurotransmitter will be released. And then you can see the different structural variations of the different classes of neurons. Here you can see multipolar has the dendrites kind of looking like a big hairdo with the cell body and the one long axon. If you ever get to actually look at stained slides of the cerebellum, I actually have stained cerebellum before. These Purkinje cells are beautiful. They are contained within a single and the Purkinje cell layer within a folia of the cerebellum, and it looks pretty cool. And then here the pyramidal cell, which looks like a typical neuron with its dendrites, and then one long axon coming from the cell body. Bipolar neurons, we can see that there are different ways of how they're structured, with one cell body and then a fused dendrite and then one long axon, and here's a retinal cell that's bipolar. And then we have a unipolar, which could be a dorsal root ganglion cell with the cell body and then the one process. This is supposedly the axon of the cell body. So neurons can also be classified functionally. Functional classification is according to the direction the nerve impulse travels relative to the central nervous system. So functionally speaking, we have sensory neurons, motor neurons, and interneurons. Sensory neurons we know are picking up signals from the body and then bringing them back up towards the CNS. These are also known as our afferent sensory type cells. Motor neurons, these are neurons that will transmit impulses away and allow for movement of skeletal muscles. And then interneurons we'll find within the CNS. So again, sensory neurons transmit impulses towards the CNS. Virtually all are unipolar neurons. Cell bodies in the ganglia are outside the CNS. And then they have short single processes that divide into the central process, which runs centrally into the CNS. And then you have peripheral processes, which will extend peripherally to the receptors. Our motor or efferent neurons will carry impulses away from the CNS to the effector organs. And we talked about the many different types of effector organs, such as skeletal muscle, glands, and such. Now, most motor neurons are multipolar neurons. These are the neurons that we looked at under the histo slides at the beginning of the semester. The cell bodies are within the central nervous system. And then they form junctions with effector cells. Now, interneurons or association neurons, most are multipolar. They lie between the motor and sensory neurons and are confined to the CNS. So interneurons more responsible for integration and processing within the CNS, within the brain. And here's a nice table with a comparison of the different classes of neurons functionally. So we have our multipolar. Multipolar neurons, most are interneurons that will conduct pulses within the CNS, again, responsible for integration and processing. So integrating sensory input or motor input, maybe one of a chain of CNS neurons or a single neuron connecting sensory and motor. And then some are motor neurons that will conduct impulses from the CNS to the effector organ or cell, such as a muscle or gland. And bipolar, all bipolar are sensory neurons located in some special sense organs. So we have them in the retina, and they will transmit their input, their sensory input, to the brain. Okay, so they carry signals towards the CNS. And then our unipolar, most are sensory neurons that will conduct impulses along the afferent pathways to the CNS for interpretation. Okay, so we have, for example, so these are called primary or first order sensory neurons. So we have these unipolar sensory neurons in our skin, and it will travel up and go towards the CNS. So neuroglia, we talked about being the supportive cells of the nervous system. There are 6 types of neuroglia. We have 4 in the CNS and 2 in the peripheral nervous system. Again, provides supportive functions for neurons and also cover non-synaptic regions of the neurons. So most neuroglial cells have branching processes and a central cell body. They outnumber neurons 10-1, which is good because the more support, the better. Neuroglia make up half the mass of the brain and can divide throughout life. So the first support we're going to talk about, the neuroglial cell we'll talk about, are astrocytes. Astrocytes are the most abundant glial cell type. They can sense when neurons release the neurotransmitter glutamate. They extract blood sugar from capillaries for energy. They take up and release ions to help control the environment around the neurons. They are involved in synapse formation and developing neural tissue, produce molecules necessary for neuronal growth, certain factors, and also help propagate calcium signals involved with memory. Here is a typical astrocyte providing support for the actual neuron. Again, these are the most abundant of the CNS neuroglia. Microglia, these are the smallest and least abundant glial cell. They can undergo the process of phagocytosis. They are phagocytes, meaning they are like macrophages of the CNS. Again, think of macrophages being that Pac-Man cell that kind of engulfs, other things. So microglia are responsible for engulfing, invading microorganisms and dead neurons. They are derived from blood cells called monocytes. We talked about monocytes being one of your leukocytes. And then these monocytes will migrate to the CNS during embryonic and fetal periods. Here we see a microglial cell being, part of the CNS defenses by phagocytizing any invading foreign invaders. Ependymal cells line the central cavity of the spinal cord and brain. They help bear cilia, help with the circulation of the cerebrospinal fluid. Oligodendrites have few branches. They wrap their processes around axons in the central nervous system and help produce myelin sheaths in the CNS. Now, oligodendrocytes are very important, especially for signal conduction along the axon. In the CNS, they're called oligodendrocytes. In the peripheral nervous system, they're called Schwann cells. Okay, so oligodendrocytes, think of an electrical cord. Inside the electrical cord, you have wires that help, conduct electricity. The rubber part kind of surrounding or around the wires within the electrical cord, you can compare that with the oligodendrocytes and the Schwann cells. They kind of form this sheath around the electrical cords to help conduct that electrical energy down the cord. Just like oligodendrocytes will kind of form that sheath around axons to allow for propagation of a chemical signal down the axon. Here we see our ependymal cells. That will line cerebrospinal fluid-filled cavities, such as the different ventricles, lateral ventricle, fourth ventricle, so help with the movement of cerebrospinal fluid. Here we see oligodendrocytes that have processes that will form myelin sheaths around long axons in the central nervous system. Okay. So here are oligodendrocytes. Their processes wrap around the axons, providing that myelin sheath. Now in the peripheral nervous system, the neuroglia that we can find include satellite cells. Satellite cells will surround neuron cell bodies within the ganglia. And then we have Schwann cells, or neurolemmocytes. Neurolemmocytes surround axons in the peripheral nervous system. So in the central nervous system, they're called oligodendrocytes. In the peripheral nervous system, they're called Schwann cells. The Schwann cells, like the oligodendrocytes, will form that myelin sheath around axons of the peripheral nervous system. Okay. So here we see the cell body of the neuron in the peripheral nervous system surrounded by a satellite cell. And then here we see axons surrounded by Schwann cells that form that myelin sheath, sort of that wrapping around of the processes of the cells to help with conduction of any electrical signals down the axon. So myelin sheaths are segmented structures composed of the lipoprotein myelin, help surround the thicker axons, again, form that insulation or insulating layer that prevent the leakage of an electrical current, also help increase the speed of impulse conduction. If we just had a normal, metal wire by itself, sometimes the electrical signal might kind of leap off without any insulation. So we put a rubberized insulation around the metal cord to prevent, leakage of that electricity. Sort of how we have the myelin sheaths. They actually provide that insulating layer around the axon to prevent leakage of an electrical current going down the axon, and also help speed up the impulse of conduction. So in the peripheral nervous system, myelin sheaths are formed by Schwann cells. They develop during fetal period and in the first year of postnatal life. The Schwann cells will wrap in concentric layers around the axon and cover the axon in a very tightly packed coil of membranes. The outer collar of perinuclear cytoplasm is called the neurolemma. That's why they're called neurolemmocytes. And then this is material external to the myelin layers. So here you can see the processes of the Schwann cells wrapping around in a circular concentric type layer around the axon. So here's the axon, and here's that myelin sheath of the Schwann cell. Okay, again, providing that insulation, help with impulse conduction speed. So we call this here, we have non-myelinated axons in the peripheral nervous system, not covered with an insulating sheath. So there are gaps between the myelin sheaths. These are called the nodes of Ranvier, and we were able to see those slides in the beginning of the semester. Nodes of Ranvier are gaps along the axon, help speed up nerve transmission. Thick axons are actually myelinated, whereas thinner axons are non-myelinated, so they conduct impulses more slowly. Again, in the central nervous system, it's the oligodendrocytes that will form the myelin sheaths in the central nervous system, have multiple processes, and will coil around several different axons. So I'm going to stop the first part of Chapter 12 here, and we will continue on with this PowerPoint for Part 2 with the gross anatomy of the nervous system.